The following program is brought to you by Caltech.
Next, it is my pleasure to honor Dr. Clyde Hutchison for his work in determining the first complete sequence of a DNA molecule and in developing site-directed mutagenesis, and for his work on synthetic genomics. Dr. Hutchison is a distinguished investigator in a synthetic biology group at the J. Greg Venter Institute in San Diego. He received his PhD at Caltech in biophysics in 1969, and while at Caltech, he studied with Professor Robert Zinsheimer, and he credits his time at Caltech as providing a foundation upon which his career could flourish. He is a member of the National Academy of Sciences and a fellow of the American Academy of Arts and Sciences and the American Academy of Microbiology. He has received the National Institute Research Career Development Award as well as a Merit Award and the North Carolina Award in Science. We are proud of Dr. Hutchison's many accomplishments and honored that he remains a champion of Caltech. Dr. Hutchison, please accept this award with our gratitude.
Well, I'm really very deeply honored by this Distinguished Alumni Award from Caltech, and I'm thankful to everyone involved, both here at Caltech and throughout my career. But first, I'd like to thank my parents for my own genome, and my father for early scientific education that kindled my passion for science. But that education had a strong emphasis in the physical sciences. And I was first exposed to biological research through a part-time job while I was an undergraduate at Yale. I had a part-time job with a young postdoc named Carl Woese, and he worked in the lab of Harold Morowitz. And as a result of this experience, I took the advice of Ernest Pollard, who was the head of biophysics at Yale, and applied to Caltech. And I still remember one day in my Yale dorm, I got a phone call, what we used to call a long-distance phone call. It was George Beadle, who was chair of biology at Caltech, and he was offering me a fellowship. So, of course, I took it right on the spot. And at Caltech, as President Chameau said, I had the good fortune to work with Bob Sinsheimer, who introduced me to a tiny bacterial virus named Phi-X174, and he taught me the power of selecting very simple systems for biological studies. And I'm also grateful because I met in his lab a postdoc named Marshall Edgell, and we formed a very productive, long-term, collaborative and friendly relationship. And we moved together to the University of North Carolina at Chapel Hill, where I was an active faculty member for 37 years. I also thank Fred Sanger, and in 1975, I embarked on a sabbatical in his lab where I took part in sequencing the Phi-X174 genome. That's the molecule President Chameau referred to. While I was in his lab, I met Michael Smith, also on sabbatical there, and this was fortunate because we made some plans, and following our sabbaticals, we collaborated to develop site-directed mutagenesis using oligonucleotides. Then later, about 20 years ago, I became interested in very simple bacteria called mycoplasmas because they have the smallest genomes of any cells that are capable of growing independently. I thank Craig Venter and Ham Smith because they made an offer to collaborate in sequencing a genome of this organism. And this project led directly to our current work on the chemical synthesis and installation of bacterial genomes into cells. So I'm grateful to all these people for their influences and contributions to my careers, and of course also my many talented and hardworking students and postdocs over the years who've been involved in these projects and many others. I also owe debts of gratitude to my wife, Pfeiffer, for many things, but in this context specifically for encouraging me to give up the shelter of academia to move to the Venter Institute, which I did finally in 2005. This has allowed me to rejuvenate a rewarding and productive scientific career, which is now in its 54th year since I published my first paper as an undergraduate with Carl Woese and Harold Morowitz. Again, I'm deeply honored and very thankful for this award.